during the presentation. After the presentation, there will be 20 minutes for Q&A from the audience.
10 years ago, to introduce our company, I would have told you a story about a cancer patient that was very close to me. I would have told you how cancer had affected their lives and the lives of those around them. Unfortunately, I don't have to do that anymore. Everyone in this room has their own personal cancer story. And if you've paid attention to the news recently, you would have seen there have been some tremendous advancements in the treatment of cancer. But what you haven't seen is an advancement in the treatment of bladder cancer, which is still utilizing 40-year-old technology. Look at a quote from a recent FDA review panel that had just rejected another bladder cancer drug. Dismal outcomes and unacceptable choices. This is the current state of the treatment of bladder cancer, the fifth most common cancer. My name is Drew McKinnon, and I'm the CEO of VivImmune, and I'm going to give you a brief overview of where we're headed today. So it's no secret that pharmaceutical drug development is a risky business. Today we're going to tell you why our solution, our team, and our strategy are set up to mitigate risk, provide a return for our investors, all while getting our solution to the patients that deserve better than those unacceptable choices. This is Sean Smith, our Chief Research and Development Officer, and he's co-inventor of our solution. He's going to tell you more about the problem and our solution to that problem.
So, as Drew said, bladder cancer is the fifth most common cancer, but it's still being treated by a 40-year-old technology called BCG, which is a bacteria that's placed into the bladder. Unfortunately, many people treated with BCG don't respond, and many others go on to have recurrence and progression, often leading to the need for full bladder removal. There's a desperate need to develop alternatives to BCG. Now's the right time to do it. Here's why. For the last 150 years, we've been limited to 3 strategies for fighting cancer: surgery to cut it out, radiation to burn it out, or chemotherapy to poison it. But recently, a 4th strategy has emerged called immunotherapy, which instead teaches the body to find and kill tumor cells. Now, maybe you heard that just on Wednesday, Sean Parker put in $250 million to start a cancer immunotherapy institute. That's because he believes, as we do and many doctors do, that immunotherapy is the future. We've developed one of these cutting-edge technologies for bladder cancer. Now, I realize that we're no Pixar, but I'd like to use this little animation to describe how our therapy works. So you can think of your immune system like a security force that's constantly screening cells. If it sees a normal cell, in this case a purple one, then nothing happens and it's allowed to pass. And any bad mutated yellow cell is quickly recognized and destroyed. Sometimes, though, they use disguises to sneak past the immune system when they shouldn't be able to. When that happens, they're able to assemble into large cancerous tumors that the immune system can't see. And at VivImmune, we work to reveal these sneaky cells to the immune system. Our lead molecule, VivImmune, consists of a cytokine, which is an immune-activating protein that raises the threat level wherever it's found. And because our cytokine, like many cytokines, works best locally, we attached it to a big sticky polymer that keeps it where it's injected, allowing us to directly inject the tumor, raising the threat level only where it counts, and prepping the immune system to destroy that tumor, and remember any disguises it finds there so that they can see through them in the future. So how does VivImmune compare to bladder cancer? Compare against bladder cancer? Well, take a look at that top line there. It's not a part of the outline of the chart. It's indicating that 100% of mice who were treated just 4 times with VivImmune were able to destroy their tumors and live a long, healthy life. And this is in accordance with our entire data package, where routinely, with dozens of experiments, we're able to destroy more than 90% of bladder tumors. To put that in perspective, if we had instead given these mice BCG, which is the standard of care, their survival would have looked no different than that control line there. We might as well have just given them salt water. And even more exciting to me is this. The only difference in these 2 mice who were given tumors on the same day is that the one on the right had previously destroyed its tumor after being treated with VivImmune. This time, though, we gave it the same tumor back. We didn't give any more treatment, but the same tumor didn't grow, meaning that the immune system was able to see through those disguises we were talking about. And this is true 100% of the time for bladder cancer, whether we try to give it back in the bladder, where we initially treated it, or somewhere else in the body. But we know that our therapy stays local through some of our preliminary toxicology data. So we've been able to generate a systemic positive immune response while limiting negative systemic toxic effects from our cytokine. So let's delve into the mechanisms just a little more for how this might work. As you may know, many tumors are already infiltrated with immune cells that should be able to recognize and destroy the tumor, but they're turned off via a variety of mechanisms. When we put our therapy, VivImmune into the tumor, the polymer portion gels up, keeping the cytokine at the site and activating the immune cells that are already in the tumor. They can then call in reinforcements and prepare for the sort of polyclonal, multivalent response that's needed to destroy the tumor as well as prevent immunoediting that can lead to escape. It also generates a pool of memory cells that can protect against recurrence. So, in this way, we're able to generate a systemic protection from a local injection. And in many ways, this differentiates us from other immunotherapies that are on the market, most of which give a less powerful molecule systemically, but they still have severe side effects, or they have to target a constrained set of disguises, making for easier immune escape. So while that data is great, it doesn't mean anything unless we have a patent to protect it. The VivImmune has been designed from the start through more than 5 years at the National Cancer Institute and the lab I work in at the University of Arkansas to be a commercialized immunotherapy. We currently hold the exclusive option to license the patent underlying this technology. It has 37 claims, a favorable patentability review, and it gives us 18 years to not only develop new molecules on the VI platform, but make it through the discovery of the development process with our lead molecule.
So it's a long way from a patent and mouse data to get into humans. And if I were to summarize the process for you, it would go something like this. There is a lot of testing, and rightfully so. We've already been through the challenging discovery process, and we've identified our lead molecule. We're currently doing small animal testing before moving into larger animal testing. And then we'll move into our targeted exit of a phase 1 human clinical trial. So we've briefly told you where we are and where we're headed. And now we're going to tell you more about the team that we have in place to help get us there.
I've known everyone on our team for about 5 years. And I got to tell you, Drew McKinnon is a natural at connecting people with solutions. He's been prepared to be our CEO of the near future through more than 8 years of new business development and medical sales. For the long term, we have targeted a more biotech seasoned CEO who has experience in drug development to help us through the later stages of our process. But Drew's going to continue on as our Chief Business Development Officer.
Sean Smith, again, is our Chief Research and Development Officer. He's a fifth-year PhD candidate in biomedical engineering at the University of Arkansas. And again, he's co-inventor of our solution. Sean also recently completed an internship at Amgen in the toxicology division. Corey Costin is our Chief Financial Officer. He is a CPA and currently is Director of Finance at the University of Arkansas Honors College.
Dr. Vanu Kovlu is a co-inventor of VivImmune and as our Chief Technology Officer, will work to strengthen our IP portfolio and build new molecules from the VI platform. Dr. David Zaharoff got a PhD from Duke, worked at the National Cancer Institute for 5 years, and at the University of Arkansas has raised more than $3 million in competitive federal grants. He's the lead inventor of VivImmune and will continue to guide our scientific direction. Now, as a team, we've thought carefully about our path to market, and we've identified 4 main risks. There's product risk in terms of scaling our solution and showing that it's safe and it works for each phase of testing. We also need to show that we're better than the stuff that's already out there, as well as do competition in the pipeline. There's regulatory risk in dealing with the FDA, and of course, cash flow. So with those risks in mind, we framed our plan around 4 key strategic decisions, each of which mitigate multiple risks, starting with our decision to target bladder cancer. So while we have that great data I showed you before in bladder cancer, we also have promising data against other solid tumors, such as pancreatic, breast, and colon tumors. But we decided to start with bladder cancer, paving the way for a later entry into those other indications for 3 reasons. First, despite the low bar set by the 40-year-old technology in BCG, there remains a thin competitive landscape for bladder cancer, with only 14 new immunotherapies in development. And if you look at the competitors that are closest to us, you see that we offer the most comprehensive set of benefits, mostly driven by a unique strategy of directly injecting a powerful molecule into the tumor and keeping it there. What this means for patients is reduced toxicity and fewer trips to the doctor, coupled with the greatest potential to teach the immune system to remember those disguises we were talking about in a way that's unique to each tumor. The second reason for targeting bladder cancer is it allows us to apply for orphan drug status. And while this provides a lot of benefits, here's what you need to know. It shortens the approval process by up to 18 months, reduces the costs associated with those clinical trials, and provides up to a 38% greater chance of regulatory approval. So we have an old technology and a thin competitive landscape, and a potential unfair advantage in orphan drug status. None of that means anything without a market. We've calculated the total addressable bladder cancer market to be about $11 billion, and And that's at a price point that's only a quarter of what current state-of-the-art immunotherapies are commanding. And once again, an entry into the bladder cancer market will pave the way for later entries into other indications, such as pancreatic, breast, and colon tumors, leading to the potential for huge market growth.
So to mitigate product and regulatory risk, we'll continue to do as much of the preclinical development and discovery work in our own in-house lab, and then we'll bring in outside expertise when necessary, specifically a contract research organization to help us with our toxicology studies, and a contract manufacturing organization to help us scale our product. Now, we talked with multiple CROs, and we selected the gold standard Charles River based on their expertise and their success with getting oncology drugs through the FDA. We've also selected Cytovance Biologics to help us scale our product, again based on their expertise and their knowledge of the different components of our immunotherapy. We've also received detailed budgets and timelines from both of these companies, and those are reflected in our financials.
So the third part of our risk mitigating strategy is to mitigate regulatory and funding risks by leveraging our scientific network and board of advisors. So as I mentioned earlier, our CSO worked at the National Cancer Institute for 5 years. We can use his relationships there to help establish a collaborative research and development agreement, or a CRADA. And while that provides a lot of benefits in terms of access to the NCI's patient-physician network and credibility around our technology, the biggest thing it provides is a substantial reduction in the cost associated with a phase 1 clinical trial. We also have an advisory board full of experts in biotech startups, new business development, and fundraising. So I'd like to highlight for you Dr. Neil Shore, who's a world-renowned urologist with over 100 clinical trials to his name. He's a substantial asset to us in terms of planning our clinical trial, as well as driving early adoption of our technology.
So the fourth part of our risk mitigating strategy is to seek an acquisition partner early. And when you think about any exit, there are a lot of things that go into determining when and if that exit happens. And in our case, we have some built-in advantages based on the market that we're in. The first advantage is that pharmaceutical companies consistently have to deal with the problem of their patents expiring. This has led to the common industry practice of acquiring small biotechs like ourselves in order to grow the size of the pipelines. The second advantage is just the tremendous growth of the immunotherapy market in general, which is the hottest space in pharma. That's evidenced by the $10 billion in deals that took place in this space in the last year alone. So again, our decision to seek that acquisition partner early is strategic. We will have assumed the risk of getting our therapy through to the clinic, but in doing so, we will have substantially increased our value, which we estimate to be $550 million after a successful phase 1. So when you consider the current landscape of the immunotherapy market, that $550 million valuation is reasonable. Now, we understand, as with any deal, there are a lot of things that ultimately will determine our value, but in our case, we consider the tremendous growth of the immunotherapy market, as well as the comparable deals that you see listed here, most of which are platform immunotherapies that can be utilized in multiple indications. As you can also see, we received feedback from Big Pharma indicating that not only is our technology unique, but also that our decision to seek that acquisition partner early is the right one. We've also been in preliminary discussions with private equity firm Bloom Burton out of Canada, and they've asked us for a data package that we're currently preparing for them so that we can continue our discussions. So how do we get to that valuation that we discussed earlier? Well, in addition to the $4 million and 5 years of research that's already gone into the development of the product at the National Cancer Institute and at the University of Arkansas, big picture, we need about $9 million in approximately 5 years to get us through phase 1. There'll be multiple exit points for us along the way. And with the $2 million in angel investment that we're seeking, coupled with the $2.3 million in SBIR grants that we will apply for, we can get through a couple of those milestones, which are proving that we can scale the product and proving that it's safe in larger animals. Now, not only will this provide us a potential exit point after only 2.5 years, but it also positions us to go after our Series A round of $4.7 million of institutional capital. With that $4.7 million, we can get through full-scale production and through our clinical phase 1, which gets us to that valuation we discussed earlier, and that's a 49x return for our angel investors. So let me leave you with this. The last major breakthrough in the treatment of cancer was chemotherapy, nearly 70 years ago. Immunotherapies are the new cutting-edge technology, and they have led to the advancement in treatment in multiple cancers. Unfortunately, those advancements haven't made their way into the treatment of bladder cancer. Someone is going to help a lot of patients and their families, and with your help, we can get our cutting-edge immunotherapy from the lab to the patients that deserve better than the unacceptable choices that they have today. We are VivImmune, and we are fighting cancer at the source.
Okay, and now the judges will have 20 minutes to ask you questions.
So there may be a trend. Oh, and thank you, very nicely done. You both look great speaking voice, slides are very nice.
Thank you.
So There is a trend, yes, and everybody's getting on the immunotherapy, et cetera, and about the immune system. But I, as an investor, want to know if you don't make that phase 1 exit, what's your plan going forward? And how much money do you think you'll need to get to market?
So, and certainly hear that concern and understand that most immunotherapies,
I'd love to know what you think it will cost you to raise, to get to market, if you don't get an exit at phase 1.
So we haven't run the numbers all the way through phase 3, but we have run it through phase 2, and we estimated about $6 million more.
and that would be continuing the CRADA as well. That's assuming that we would continue our CRADA agreements.
All right, thank you.
According to my understanding about it, in this area in Norway, that immuno-oncology is hard, but there are different base technology. We have antigen, antibody, development base. And in 2014, GSK dropped one of their immunotherapy oncology program after they spent a lot of money there. Is your technology similar to the GSK technology or different technology?
I'm not sure exactly which technology you're referring to.
The MAG-A3, MAGE-A3,
Oh,
that is a GSK.
okay, yeah, so I believe that that's a cancer vaccine.
Yep.
And so what it does is secrete a particular number of antigens.
Right.
But, I mean, one of the problems with that is you can't ever know how many antigens you need to go after. That's one thing. And another thing is it doesn't really potentiate a strong response. So it's sort of passive in the way that it works. So it is a different technology.
Different.
It's different.
Okay, thank you.
Sure.
Is there a veterinary market for your technology that would help you with your go-to-market plan costs?
We've actually talked about that. One of the animals that does get bladder cancer are dogs. So there is a potential earlier entry into the market through the veterinary space. We weren't as familiar with how to get it into those animals, like what the regulatory process was, but that's something that we've tossed around.
Can you tell us again how your technology works and tell us a little bit more about your animal study?
Yeah, sure. So, like, we have our, let me go back to that slide for you. There's a, there you go. So, what we do is we have that cytokine which talks to the immune system, and it's attached to a polymer that's quite a bit bigger than what's actually on the screen. And what that polymer does is, when it's outside of the body at a pH of about 6.5, it's liquid. It's viscous liquid, but it's liquid. Put it in the body when the pH is higher, and it gels up, trapping that active site where we inject it. And so that can then turn on immune cells that are at the immune site, which are turned on. And then they can call in more immune cells that can help destroy the tumor and then generate a memory response.
But you're
Inventory coverage.
Yeah.
Yes,
And indirect.
all your
And
offices.
so what does the intellectual property cover? So it's covering the polymer, the method of use, or what's the intellectual?
It is covering the way we attach these two things.
Oh,
And then especially with this polymer on that side.
Who's got the IP on the polymer? What's
It's,
it called? It's commonly known polymer.
yeah, it's common.
The cytokines.
But we have to modify it a little bit in order to make it work.
We're modifying the polymer.
The polymer and
And
the
the
cytokine.
cytokine.
Yeah, because as you might know, if you just attach the cytokine anywhere, then you can lose activity.
Yeah, right. And then you get some sort of some other response. So,
I think Deborah had her.
Well, just to continue on the slide here, so whenever you inject this near or into the tumor, do you have any data for killing stem cells in the center of the tumor?
Cancer stem cells. We haven't looked at cancer stem cells in particular, but we also haven't seen the tumors come back. So,
So the answer's no yet.
no.
So how many animal studies, you didn't finish the question on the animal study, how many animal studies have you actually done?
I actually don't know how many we've done. I know that I'm up to bladder 33. So that's something I've done.
Previously, you just mentioned that you were gonna have to modify it to make it work. Can you expand on that comment that you just made?
Yeah, sure. So that's actually something that's in our IP and something that makes us sort of unique. So if, I'm not sure how familiar you are with proteins and how they work, but in order for them to bind to a receptor, they need to have their binding site open. And so you can imagine that if we accidentally bound the binding site of the cytokine to the polymer, then there would be access to that receptor. So we chose sites that were far away from that.
Does your polymer technology has time release capability?
Well, it doesn't allow, that's something we're still characterizing.
Okay. So is it hydrogel based or not?
It turns into a hydrogel when it goes into that.
Okay. Because hydrogel does have time release capability.
It would be if we were just co-formulating them together. And with this, it's a biodegradable polymer, so it will degrade over time within about a
Right.
week. And it would rise.
Right, that's the property.
Yeah.
So it's not like you're not getting inside the cell, you just attach it to the receptors.
Yeah.
Okay. But from molecular said that the key is to try to get inside the cancer cell. Right? If you can get inside the cancer cell, would it be your molecule, would it be more effective?
It wouldn't work because,
It wouldn't work.
because receptors live on the outside of the cell. And so we have we bind on the outside, and that sets off a chain of reactions that move inside the cell. And we're not targeting cancer cells, we're targeting immune
So
cells.
you feel right.
And so then the immune cells actually latch on and kill the tumor cells.
Okay, thank you.
So going back to the business, so since you are pretty bullish on a phase 1 exit, how warm are you to a strategic partner that may be interested in funding some of your studies or partnering with you? So is it, we're thinking about it, we've talked, or we are almost dating? Thank you.
So, we have had conversations with Ashton and VivImmune that said they wanted to see some initial pre-tox data. Because they said, we've done deals where you're at right now, but we would like to see, because we haven't seen cytokine this close to the clinic, we'd like to see some pre-tox data, which is what Sean is currently working on in the lab, so we can go back to them. And at that point, they would start their due diligence process and we go down that line. But we're certainly open to strategic partnerships.
Have they veilingly suggested anything?
No, not at this point. They want some initial data.
So what is your strategy to deal with strategics, please? And when?
When to go after them? We don't necessarily have a strategy to go after strategics at this point.
Do you have a plan for leadership to go after them?
That would be, so we're hiring, our plan is to hire that full-time biotech CEO 12 months before our Series A round, 4.7 million. And that would be his full-time role,
Do you think you need a full-time or a part-time?
full-time.
Thank you.
If you win a bucket of money here, would you be willing to move your business to Houston?
I think all options are on the table.
The answer is yes.
Yes.
Right there.
I was gonna say, right this second.
What kind of documents do you have in place before you start conversations with potential strategic partners?
We signed a non-disclosure agreement with them.
So say again what your licensing status is, you said you had an exclusive option or you have an exclusive license?
Well, we had the exclusive option, and that was set to expire in June, and we can renew it at that point. It was set on the timeline.
Was this an option with terms or an option without?
It's the exclusive option to license it. It's 1% of the liquidity event that's non-deliverable. We have to pay off all the rest of the patent costs and the costs of already incurred. And 1% of market royalties, but based on our plan, we don't expect income, so that should be fine.
So once you actually execute a license, you'll look in the patent law.
Correct. Sir?
What sort of challenges do you expect in manufacturing the product? Do you kind of acknowledge, what it looks like to make this day in, day out?
Well, I think you're right to hone in on that. That biologics can be tricky to make, but we've gotten much, much better at it. And especially the CMO that we talked with is much, much better at it. And we also have a GMP source of polymer, and that'll act as a supplier to them. They should be able to congregate them. Yes.
So you'll contract this out?
Yeah.
Contract out manufacturing?
Yes, sir. That's our plan.
Who was your contract manufacturer?
Saturdays.
So you said earlier that you guys have been working on this molecule for 6 years, or 5, 6 years.
Mm-hmm.
How did the idea of this molecule come about?
Yeah, well, my PI, he did his postdoc at the National Cancer Institute.
Okay.
And while he was there, he tried a variety of different cytokines. He says, wow, this one works the best. But in order for it to really work best, we need to keep it local. Because that's how the body uses it. So let's use it like the body uses it. And so he started just mixing these two things together and iterated into this, which is now the matching.
Okay. Thank you.
So there's no toxicity associated with localized application, is that it?
Nothing we've seen in mice.
Is the technology owned by another?
No. No. I'm not.
You said it was invented while PI was there.
That's where we started thinking about it. It was invented at the University of Arkansas.
Invented and active use of life.
We had another question back there.
I have the same question,
Okay.
because whenever you said NCI, I wanted to know what the ownership to the university and then to y'all was. Because I similarly am concerned about that if they know something goes forward I would look into this. I'll do it in feedback, don't worry about
Okay,
it.
thank you. Yes, sir, we have one over here.
Yeah. Data, I assume you have a lot of data based upon your small animal study.
A lot of data?
Yeah,
Yes.
reports. So a lot of reports. Good.
Publications?
Yeah.
Yeah, so we got 11 publications on the different components, but not of the actual one.
You have in vitro studies showing activation of the responsible cells. What cells
Yeah, yeah.
specifically are active?
Yeah, so we use this, we actually have verify the bioactivity after we put them together. So the exact mechanism we're really still figuring out, but we do know that there's an increase in CD8 cells in the lymph nodes, there's an influx of macrophages. We know that if we knock out CD8 cells, then the therapy won't destroy that initial tumor. We know if we knock out CD4s, it will still destroy that initial tumor, but it's the opposite on the rechallenge. So if we knock out CD8s, that rechallenge will still ungrowth. But if we knock out the CD4s, then the rechallenge will grow, which is sort of counterintuitive. But it's so the helper cells play a big role.
Increase in macrophages and increases.
In the chart that shows the control group of mice versus those that, received the therapy, how many mice have been tested?
So in that particular experiment, it was about 10. But we've done it through dozens of experiments, and I would say on the order of 100. Well, mice live about 2 years, and we re-challenged them 18 months out. And they still haven't grown.
And so you're 90%, you're 100%, or across all of the studies, or
Yeah.
a couple of studies?
All the studies. So,
In that graph, you said that, for the control group, you gave them BCG, you might as well not give them anything at all.
yeah.
Does that mean that the BCG is not effective at all in any of the mice?
Yeah, so what we were trying to model there is sort of a worst case scenario in which BCG doesn't work, which happens in 30 to 50% of patients who still go on to experience recurrence. And so that's really our initial target market. BCG eligible patients. Any more questions?
Can you comment on shelf life and storage conditions?
We're not there yet.
Are there different kinds of bladder tumors, and does that affect the efficacy?
Different stages.
Or even types, types of stages with different types?
Yeah, so there's a variety of stages. Actually, I have a slide for that. There it is. So, most bladder tumors are caught in the T1 through carcinoma and C2 stage, which is kind of early stage. Like 70% of tumors are caught really early. Mostly because one of the ways you know that you have a problem is you start seeing blood, and that'll get you into the doctor. There's different stages, and the stages that BCG treats is T1, which is, it pokes out into the bladder. And so what they do right now is they go in and they scoop some of it out, and they stage it. And if it's high grade, they give BCG. If it's low grade, they just give chemotherapy as a wash in the bladder.
So you go in and you shoot it.
That's exactly right.
Okay, time is up. Thank you.